So thank you so much, Giovanni, for the introduction. Very warm welcome to our students and colleagues at King's College London, and also to our colleagues at the other London colleges and universities, and to the world at large. Our registration shows that we have up to 600 registrants, and I welcome you all for this exciting day. The morning will be spent mostly with reports on the progress of a particular grant we obtained from King's. And the afternoon will be keynote speakers, also from Europe, and a Nobel laureate later in the day. So we are looking forward to a very, very exciting day. My introduction goes a little bit back in history. At King's College London, Professor Heide, who was heading the iron metabolism group for a very, very long time, at least 20 years, key work in the field of iron metabolism was done at King's, the cloning of many iron transporters, and there is still a very, very active interest. But we branched out a little bit to what is now the metal metabolism group, which is a group hosted in the Department of Nutritional Science with Krista Hoxton, Paul Sharp, Yemisi Latunda, and myself, but also involves many, many interactions with others in the school. And as Professor Mann mentioned, we have a London Metallomics Consortium where interested parties from the London universities meet to plan a symposia, grant applications, and discuss progress in the field. And the last aspect is the London Metallomics facility, which is part of the symposium and is actually supporting this symposium. It was supported or is supported by a Wellcome Trust equipment, which KCL received together with Imperial College. And it is heavily funded by King's College London. And we want to thank at this point King's College London for this incredible support we had over the years. Now, the symposium was put together because we obtained that King's Together Strategic Award. So that's a King's College London award. And I have listed all the people that contributed in one or the other way. There are external partners, there are co-investigators, and you will meet some of them during the day, or you will hear about their contributions. I haven't told you what metal fingerprints are. That is the focus of our day today. Metal fingerprints are specific metal signatures, metal compositions of cells and tissues. And I think that's one of my first major messages. The metals are not randomly distributed in your body, and there's very active metabolism for their distribution and their control. And that's the subject of what we're going to discuss today, because it is a fingerprint of whether or not a tissue or a cell is healthy or is diseased. So it has a bright future in terms of the application for diagnosis of disease, for disease mechanisms, and also finding out how we can treat various diseases. And in many cases, the perturbation of metal metabolism is not only a consequence, but a cause of the disease. So I'm not back at full screen. Let me see. Okay. So sorry about that. So what is metallome and what is metallomics? The term metallome was actually coined by the late Archie P. Williams in a key paper in 2000, and it is the assembly of all the metal species in a cell. The term metallomics was then introduced by a professor at Nagoya University with the name of Hiroki Haraguchi in 2004. And it blends very well into the other omics fields, and that's what the slide is telling you. You have genome, proteome, lipidome, glycome, and you have the metallome. So it is well established now as the field of metallomics. And that is something the London Metallomics facility is focusing on. A real game changer in the history of metal and element analysis were the different spectroscopic and spectrometric techniques, which increased the range of detecting metal ions enormously. Before that, it was mostly rather insensitive procedures, and only these new techniques opened the window for the understanding of metal metabolism and the metal distribution in different species. Flame atomic absorption was a game changer, and then a version that is graphite furnace AAS. But what really brought another step in the field was the development of inductively coupled plasma mass spectrometry. And you will hear about this more today, which increased the sensitivity enormously. And we can analyze some elements and some metals at sub-part per trillion concentrations. And here I showed you I'm showing you from Professor Haraguchi's work what the distribution in a living species is. This is salmon eggs, but I will switch to humans in the next slide. So all the way from milligrams per gram metal ions, micrograms, nanograms, and picograms, and then we can even measure concentrations that are lower than that. In red, I have shown you some elements that are not essential for humans. And the point of this slide is that some elements that are not known to be essential for us are occurring at higher concentrations than essential metal ions. Let's take in the fourth row molybdenum. That's much lower than, for example, rubidium or arsenic or barium or nickel. So many of these elements are present in your body, but we do not know exactly what the consequences are. And that's something which we are exploring and which is very, very important. The concept of trace elements came because the trace elements here listed at the bottom are less than 1% of your body weight. But this is really a cutoff that is debatable. What I'm saying here is that, for example, zinc is 2 to 3 grams and magnesium is about 19 grams. Yes, it is about 1 order of magnitude lower, but the importance is not really shown in this slide. So quality and quantity are really two different things. So referring to some of these elements as iron, like iron and zinc as trace elements, is putting some question on what they are actually doing and how important they are. Now, in this slide, I show you all the elements, essential nonmetals and essential metals that are important for humans. The ones circled are important for other organisms, showing you that a large part of the periodic system of the chemical elements is actually important for life. And that is, again, a very important take-home message, because we concentrate on our focus with proteins and lipids and sugars, mostly on the elements like carbon, hydrogen, oxygen, and nitrogen, sometimes phosphorus. But all these elements are essential and very important for you to survive. Coming back to one of the previous slides, this is now the periodic table that shows you all the elements that can be measured with these high sensitivity modern techniques. And you will see that we have a lot of lead, for example, Pb, almost the same concentration as zinc in our bodies. And many of the elements here are not known to be essential. You even have uranium in your body. You can measure that. So many of the elements that are important for some function, not necessarily essential functions, but we want to understand what the implications are if by any reason you have a higher amount of any of these elements. Now, I will spend a couple of minutes on introducing you to zinc, which is the element which we are interested in at the moment, and I have been interested for many years. But it has come a long way because it was very, very difficult to detect. And only recently, let's say the last 20 years, we are starting to understand what the importance of that element is. And as I indicated earlier, the concentrations are almost as high as those of magnesium. And everybody knows that you need magnesium, but the knowledge about zinc doesn't go far beyond that people know that they need it. And I will say a little bit about that. So 2 to 3 grams in a human body. The concentration in the cells, however, is very, very high. You have 0.2 millimolar zinc in human cells, and compare that to the concentration of a very important metabolite like ATP. The average concentration of ATP is somewhere in the range of 1 millimolar. So you have almost stoichiometric amounts of zinc compared to ATP. So it's a lot of zinc. And then if you go to subcellular organelles, compartments, the zinc concentration is even higher. So up to 10 millimolar. Or in some compartments, depending on how you count it, it can be approach even up to 1 molar. So very, very high concentrations of zinc. And I will tell you a little bit why that is important. And one last note, when you look at health stores and advertisements, you will frequently see that zinc is sold as a so-called antioxidant. Well, how can it be an antioxidant? It is always zinc 2+. It is not redox active. It is the redox inert in biology. So the reason for that is that it has sometimes some indirect antioxidant actions by the genes. It is involved in gene expression, but that really is a matter of concentrations. If you ingest by any reason too much zinc or have conditions where your zinc accumulates, you can have the opposite effect, that you have indirect oxidant actions. The next important slide here is to show you what the scope of zinc biology is. We have over 3,000 proteins that require zinc. And that's not even including all the enzymes that are regulated by zinc. If you translate that into what we know about the expressed human genome, every 10th protein is a zinc protein. And that shows you how important that element is for various functions, namely at least 1,000 enzymes with the catalytic function depending on zinc. It is a structural metal ion, important for making the three-dimensional structure of proteins. And for protein-protein interactions, where it can bridge proteins. And then it has regulatory function, which are just being recognized right now. And you may have noticed that I'm only talking about proteins. Most of the biochemistry of zinc is linked to proteins. We know relatively little about interactions with other biomolecules. If you compare zinc metabolism and control of zinc homeostasis with iron homeostasis, there is again an important factor that you need to note. We essentially have one iron importer and one iron exporter. If you look at the zinc field, you have 24 transporters that coordinate the control of this particular metal ion in cells. Transporters that bring the zinc in on this cytoplasmal membrane, and many transporters on organelles. So that has to be highly organized. And then in addition, you have at least a dozen metallothioneins that participate in the distribution of zinc. So think about this, just for this one element, we need in the order of about 50 proteins that are just involved in control. And that again shows you how important that element is. Now, I talked about regulatory function. There has been a change in the field which goes back to about 20 years, but it's now recognized widely that zinc ions are signaling ions, and they complement calcium signaling. Calcium signaling is somewhere in the nanomolar to micromolar range of concentrations. Zinc signaling is below that, in the low nanomolar range. So zinc signaling interacts with calcium signaling, and both influence redox signaling and phosphorylation signaling. So it's a central theme that affects many signal transduction pathways. How do these zinc signals work? Well, similar to calcium signals. There is release from intracellular stores of zinc ions that then affect and regulate proteins. And there are many examples. And you need to look at the literature on that. I can't go into this in detail. But there's also extracellular signaling. So many cells actually secrete zinc ions by exocytosis for a purpose. And that's cell signaling, cell-cell signaling, and there are many, many examples for that. A very exciting field. And I will, in a few slides, show you what our present interest is, and then come back to how we apply metal imaging in the field of zinc as an example. So we have been interested in diabetes for quite a while, and I'm only focusing on that. We're interested in arthritis, skin conditions. I have been interested in liver disease, neurodegeneration, and with my colleague, Krista Hoxton, we are interested in cancer biology, where changes in zinc have huge impact on cell proliferation and cancer progression and metastasis. Now, back to diabetes and glucose metabolism. We have been focusing on two aspects, namely zinc in the beta cell, where you need millimolar concentrations of zinc to store insulin. The zinc is actually secreted with the insulin. So that's one aspect we are interested in. How does that transporter work that brings zinc into the insulin granules? And then the second aspect is that we need zinc in the cells for various purposes. And we have been interested in the control of protein tyrosine phosphatase 1b. So one or two more slides, and then I'm done. This is a letter from the White House, how President Trump was treated. And it is clearly stated that he received a treatment that included zinc. And I think this is very, very important because I have heard relatively little in the discussions at the universities and in the discussion in the public domain about the importance of zinc. I wouldn't go into it, but there are clinical trials running in several countries that zinc has a very, very important effect on the progression of the disease. And I leave the rest to you. I just want to acknowledge the collaborators, Krista Hagstrand in particular, my colleague, my past postdocs, and the Department of Nutritional Sciences, and then the granting agencies that have supported our work. So I will close now to leave enough time for the others. I want to remind you that there is a booklet available online that describes our work, gives summaries of what we are doing, and biographies. So I invite you to look at this, and I wish you a wonderful day, which you hopefully will enjoy in terms of the really exciting new research that is being done in our facilities, but also in others in the world. Thank you so much for your attention.
Thank you, Wolfgang. I don't know if I'm live. Yes, you can hear me. Wolfgang, thank you very much